Okay, so the next talk will be given by Dr. Marco Palombo. Dr. Palombo is an associate professor in Cardiff University Brain Research Imaging Center in the School of Psychology and the School of Computer Science and Informatics at Cardiff University in Cardiff, UK. He holds a physics degree and a PhD in biophysics from Sapienza, University of Rome in Italy, and worked as postdoc and senior research associate at the Molecular Imaging Research Center in CEA in Paris, and also at the Center for Medical Image Computing in Department of Computer Science in UCL in London. He's the recipient of a Future Leaders Fellowship from UK Research and Innovation Council. And today he will be talking about the gray matter, modeling and validation. So, Dr. Palombo, the floor is yours.
Thanks for the kind introduction. I'm going to share my screen and start the presentation. Do you see presenter mode or?
We're seeing presenter mode, so if you can switch it.
Yeah, 1 second. Where is it?
Maybe in display settings at the top.
Cannot see that. Oh, okay, here it is. Okay. It was hidden. Now you see correctly, right?
Yes, that's great.
Okay. And you see my pointer?
Yes.
Okay. So sorry for this technical delay. Thank you so much for the invitation to give this talk today. It's with great pleasure I accepted because I really like what you guys have been doing with QMRI virtual conferences, and I'm a big fan of yours and a great supporter of the initiative. So I'm particularly happy to be here today. The topic I was asked to talk about is quite challenging itself because it's about modeling in the first place, gray matter, and then second is the validation. So I already can tell you there is not much modeling, not much validation in the literature yet in the gray matter when it comes to quantifying gray matter microstructure using diffusion MRI. And the reason is because it's quite a new topic, and it's just in the last probably 5 years that the community has focused more and more onto this. And so today, the talk will be about what has been done so far and what we have learned in our journey in the last probably 5 years, although you will see people started looking into gray matter already in the early 90s. So Riccardo did a great job already describing what models are and what is essential to build a good model of any kind of biological tissue. So models are sketches of the reality, and for a model to be a good model, it has to capture the most meaningful feature of the tissue you want to model. Now, when it comes to white matter, the previous talk from Riccardo did a fantastic job in showing you what are these features. But the question here is, what about the gray matter? The gray matter is incredibly complex and is much more complex than white matter. You can see already here from this wonderful reconstruction from electron microscopy of what I'm talking about. In the gray matter, you have blood vessels, you have neurons and glial cells, you also have epithelial cells, and the cells themselves, they are very complex. They are not just made of fibers or neurites. They are also made of cell bodies, and the neurites are not, if we want, as simple structure as the axons. They can have protrusions, which are called dendritic spines, and they very often undulate and curve on themselves. And this is true for neurons and glial cells. So answering this question is extremely difficult for gray matter, and that's why the modeling field is still a very interesting open field and vastly unexplored. In my talk, I'm going to focus on first the microstructure, and so I'm going to do a quick review, a brief review of what we know about the gray matter microstructure and what we know about the microstructural features, all of them. And then I'm going to narrow it down, going into the biophysical modeling, try to understand what of these hundreds of interesting features that characterize the complexity of the gray matter, which of them we can probably measure, and how we can estimate, and how we can probably estimate them using diffusion MRI, what has been done so far about that. And I'll conclude with some take-home messages. So let's start having a quantitative view of gray matter microstructure, and I'll start subdividing first the overall tissue, and then looking at how the extracellular space look like and how the intracellular space look like from the microstructure point of view. So if we take the human brain, it is comprised of billions of cells, roughly 100 billion of cells. And if we look at the gray matter composition in volume, the vast majority of the volume, more than 70%, is occupied by the intracellular space. Roughly 20%, depending on the brain region we are looking at and the species, is occupied by the extracellular space, and about maximum 5% is vasculature. If we look at the intracellular space, approximately 20% of this 75% comes from the cell body, and the rest is mostly cellular projections. If you look at the old cells, I'm talking about cells, not neurons. If we look at all the cells, we can divide them roughly into neurons and glia. And the ratio between these two different cell populations is different. It's not 1-to-1. So on average, in an adult male brain is 1.4 glial cells per neuron in human. If you go to other species or you go male or female, we have differences. However, roughly we are around 1 to 2 glial cells per neuron. And if we look at the glial cells, what is their relative contribution, relative presence in terms of cell count, we have mostly oligodendrocytes in number, and then followed by astrocytes and microglia. It's hard to convert this estimate into volume fraction occupied by these cell types, because they have very different morphology and depends very much on the species. But these numbers actually would reverse if we look at the volume fraction. And with astrocytes occupying most of the volume, they are the biggest between the glial cells, followed by oligodendrocytes, and then microglia occupied probably around 10% of the volume in general in the gray matter, in the cortex at least. So if we look at the extracellular space, it is a very tortuous space, as you can see in this image, and it is extremely challenging to assess. So far, electron microscopy techniques have shown us images like the one here, where the extracellular space is so tiny and tight around the cells to make us think it's mostly negligible. However, these were techniques, old-style electron microscopy, that were shrinking the tissue. Nowadays, we have cryo-EM, for example, which preserve much better the extracellular space, and we realize that the actual extracellular space in the gray matter is actually non-negligible. It's between 15 and 30%, depending on the region and the animal species. Also, the width of the extracellular space, it's between 30 and 40 nanometers, and it is not a regular width. You have highland and regions where it's much tinier, and regions where it is much more width. the width is bigger. And within the extracellular space, we also have vasculature, as I said, between 1 and 5%. And the average distance between one neuronal cell and a microvessel is about 20 microns. Although they are not that dense in the tissue, it's actually quite easy to find a vessel near a neuron. So they are everywhere, basically, although they occupy a small fraction. And if we look at the geometry, at the complexity of the extracellular space, this is highly tortuous medium. So the total tortuosity has been estimated to be between 2 and 3. And with total tortuosity, we mean not only the geometrical one, how tortuous is the path to go from point A to point B, but we also mean if you have a molecule like water, which is electrically charged and can bind to protein and molecules along the path, how much this tortuosity, these interaction effects had to the tortuosity. And we end up with a value between 2 and 3. However, if we look only at the geometrical tortuosity, the ratio between the direct path between A and B and the actual tortuous path, we have a factor 1.5. It's hard to link this to the diffusivity, but we can say that roughly this geometrical tortuosity can explain the observed water diffusion in the extracellular space to be between 1 and 2 squared micrometer per millisecond. Also, how is the extracellular space? Is it anisotropic, like we think in the white matter, or it is isotropic? There are not many studies investigating this. The only one I am aware of, and it's quite recent, is from Vincent et al., which used the diffusion of sucrose, which is a molecule only in the that is not uptaken directly by the cell, so doesn't cross the membrane, is released in the extracellular space and can be tracked with diffusion spectroscopy. And this study showed that the extracellular space, at least for the diffusion of sucrose, is isotropic and is also not Gaussian, of course, because it's very tortuous. So we can basically conclude that the assumption in our models that the extracellular space is isotropic and is characterized by an effective isotropic diffusivity slower than the free water is actually a good model assumption. Now, if we look at the intracellular space, again, it's mostly fibers, and then it's subdivided into cell bodies, which we will call soma, which are both neuronal and glial cell body. And then if we look at the fibers, we have dendrites, 45%, and axons. Of these axons, which is only 30% of the whole fibers in the gray matter, only 5% are myelinated. The other are unmyelinated axons. And most importantly, the myelinated one, they are not myelinated continuously. They are myelinated in batches. And the size of this batch of myelination very much depend on the cell type of the neuronal type from which the axon is radiating. And in this paper, you can see how they measure the proportion of total axon myelinated as a function of the different cell type, but also how long are these unmyelinated segments. And they depend on the cell type, with an interesting fact that most of the unmyelinated segments are less than 5 micron long. So you have this 5 micron non-myelination, then myelination, then non-myelination. However, a non-negligible fraction of them can be as long as 150 microns. When it comes to the cell bodies, in the gray matter, we said that is mostly neuronal, 20%, and glial, 5%. But these numbers change with age. And in fact, with age, both population, both fraction of cell bodies increases, but also in some cases, they flip. And you can end up having more non-neuronal cells, more glial cells than neurons. So it's very, very difficult to quantify all of this. And what is even more difficult is to gather together the actual values for the microstructural features. So if we look at these different cell types in the brain, we can clearly see they have all different morphologies. And if we focus on the fibers, we can see that the fibers of the granule cell is quite different from the fiber of an astrocyte. And likewise, quite different from the fiber of a pyramidal cell. However, How different are they? What are the numbers? So this was something that we always wondered, and a couple of years ago, we embarked in this effort to characterizing this fine microstructure of the cells from 8 cell types, 3 animal species, using openly available data set, the Neuromorpho and the Allen Cell Atlas. And in the abstract, which is a work in progress, will become a paper soon, you will see the description of the features we quantified that we believe are interesting for the modeling of the gray matter, with a thorough characterization of them, subdivided into cell type and animal species. As a summary here, you can see we have divided them into neurons and glial cells. And you can appreciate how very different are some of the features and how very similar are some other of the features. So this already tells us a lot on our ability to differentiate neurons from glia using only one technique, such as diffusion MRI, that doesn't contain cell type specificity in the way it probes the microstructure. So for something, for example, the cell body, it could be possible, potentially, to differentiate what cell bodies are neurons from what cell bodies are mostly glia, because they are quite different in size. But if it comes, for example, to the size of the fiber, they are all more or less within the same range. As well as, for example, the surface to volume ratio or the microscopic orientation dispersion, which is this parameter macro D. So in summary, what we have learned from this investigation is that if we look at the overall cell domain, not only neurons, I'm talking about on average within a voxel that contains neurons and glia, we would expect to measure cells with an average of 6 projection radiating from the soma, and an average overall size of the cell on the order of 300 micrometer, with the soma on average occupying 30% of the total cell volume, not 30% of the voxel volume, but total percent of the total volume, intracellular volume in the cell. If we look at the cell body, is on average 7 micron in size, and it is quite connected to the branching, branching structure, to the fibers. In fact, 18% of the surface on average is open with these openings you can see here, and allows water to flow from the cell body to the dendrites, or from the cell body to the cell fiber if it is a glial cells. And then the fibers have an average length of 50 micron. The sides, in this case, the radius is about 1.5 micron, so much bigger than the axonal radius. And they are very tortuous, 1.3. And they have also they are also curved. So the overall curvature is 60 micron. So they are not very straight fibers, but they are not even closing on themselves. 60 micron is quite a big radius of curvature. So it's pretty much what you can see here. So as I said before, this also suggests that some features, such as the branch length, does not allow us to use water diffusion MRI to differentiate between microglia and neurons, or between glial cells and neurons. But other features, for example, the soma size, because the cells naturally cluster into regimes where their sides are quite well defined, might be possible. So from a multi-compartment model point of view, what of these features can be actually measured with diffusion MRI in a reliable way, and which one cannot? So as we have seen, first of all, most of the space is intracellular in the gray matter, and most of the intracellular is fibers. So the first feature we can imagine we can measure in the gray matter is the fiber density, or the neurite density, if you like. And so, in fact, the first model introduced for gray matter in the early 2000s used only straight cylinder as main feature of the gray matter. let's assume you have access to a very powerful scanner, either a preclinical scanner or a connectome like the one we have in CUBRIC, and you can push your acquisition to be a short time regime, so short pulses and short diffusion time. What of the features we mentioned can make an impact in your measurement? So the cellular projection curvatureness is likely negligible. We say this on the order of 60 micron, which means your diffusion time should be very long to be able to appreciate its impact on the measurement. So the curvatureness of the fibers can be neglected. The branching can be neglected because the length of the fiber, again, is about 50 micron. So you need a longer diffusion time to start appreciating the finiteness of the dendrites, for example. And the projection ondulation is also negligible, given that we have measured the microscopic orientation dispersion, and according to these works, the impact is pretty negligible. So we can conclude that the assumption that we can model a fiber in the gray matter as a straight cylinder, infinitely long, not finite cylinder, is pretty much of a good first approximation based on what we know now. Back in the days, in the early 2000s, Sune Jespersen and collaborators proposed the first biophysical model for the gray matter, where indeed they proposed the intracellular space to be modeled as diffusion inside cylindrical compartments with a given orientation distribution function. So, and the model is pretty much schematized here, where the complexity of the cells is simplified as a bunch of randomly oriented cylinders. And this model worked pretty well when it came to characterizing and estimating the fraction of myelinated neurites in the gray matter. Here you see the correlation between the estimated signal fraction from the cylindrical compartment and the estimated myelin intensity in the area. And here you see a visual comparison of what is the map they obtained with the model and what was the histology. And also, because the orientation distribution of the fiber was a parameter, they were also able to show that we have sensitivity to it in the gray matter. So we can measure, this is from diffusion MRI and this is from the histology. And you can see we can measure correctly with some degree of uncertainty what is the directionality of the fiber in the gray matter. So we can also say that what happens instead, if you do not have access to powerful scanner, if you work more on a more clinical scanner or below, let's say, the connectome, if we keep the connectome as a reference for human acquisition. And so if you work in that regime, of course, you lose sensitivity to the diameter of the fiber. And so you move from a modeling fibers as cylinders to modeling fibers as sticks that Ricardo already explained in the previous talk. Now, what is the cool feature of diffusion inside stick? Is that if you go at very high B value, the signal functional form is very peculiar and characteristic. Scales, the signal at high B scales like 1 over square root of B. So as a form of validation, in this case, of this model assumption that the fiber can be approximated as a stick, one way of validating this is to just push your B value very high and see if you get this characteristic scaling with the B value. What happened when people has done this, especially in the human, is that this is not the case. So it's very much the case in the white matter, where if you push the B value, you reach this regime where the signal scale like 1 over square root of B. But in the gray matter, you break this, let's say, functional form. So this was already shown in 2017 and has been replicated in multiple studies. Now, the question is why? Does this mean that the stick assumption is wrong? Or does this mean that we are missing something else in our model for the gray matter? And this is exactly what follow-on work investigated. So part of the community start looking into including the cell bodies as possible extra feature to estimate. And part of the community is focused on looking at, okay, what if these sticks are actually not impermeable, but water can exchange? So I will go very quickly through this because I'm already 3 minutes to the end. But pretty much the first assumption is our model is missing an important feature. And so that's what one of the first work in this publication called Sandy was proposing to add this feature into the model. As I explained before, after the fibers, the cell body is the second most important feature in the gray matter in terms of volume fraction and in terms of sizes. And so we could potentially measure it. So to my knowledge, the first model proposing explicitly this is the Sandy model, which with numerical simulations have shown that indeed the signal, if you work at high p-value regime, you can disentangle the signal coming from the cell bodies from the signal coming from the neurites. And this has been also validated with numerical simulations later on. And you can get parametric maps of soma signal fraction and neurite signal fraction. Here we can see a comparison with the myelin staining. Again, in purpose chosen myelin staining, because the idea here is that the sticks are impermeable, and so most likely this model assumption hold for myelinated neurites. And then you can obtain a cortical contrast in cell body signal fraction that should mirror the cell body density. In terms of validation, one work has validated the correlation of the signal fraction in the cell body estimated with Sandy with the Allen Brain Atlas of the cell body density. And more thorough validation in ex vivo is work ongoing, where we tried to focus on the barrel cortex, where there is a clear organization of the cell bodies with a clear layering, and we have done Sandy and histology on it, and we have estimated the profile across the laminar structure with Sandy and with histology, and we found a very strong correlation, as well as we found a good correlation between the estimated values of the cell body diameter from Sandy with this one, and the one estimated with histology. This is still work in progress. It was an ISMRM abstract. Finally, the exchange hypothesis was initially proposed by Berard in one ISMRM abstract in 2018. And they're proposing a modification of the stick model, which include the possibility of exchange between the sticks and the extracellular space. They showed that this was able to characterize the breaking of the functional form at high p-value and obtain a first estimate of exchange time in the gray matter on the order of 10 to 30 milliseconds. Follow-up works by Oles and Angelescu, they extended that framework, and they also into what is called the Nexi and SMAX models. And both of them have demonstrated that they can characterize the time dependence of the diffusion signal in the gray matter, and estimated exchange time with Nexi are around 20-40 milliseconds in the rat cortex. This has been tried to be put in validation setting with histology. However, validating exchange time is very difficult. And so the hypothesis done here is that glial cells are more permeable than neurons. And so if the exchange time should correlate with, for example, astrocytes staining, GFAP, which was found, there was found a positive correlation. If you use SMAX, the exchange time estimated in ex vivo is around 5 milliseconds. So here, these studies show that it's important to account for exchange, and that in vivo and ex vivo, when you model gray matter microstructure in vivo and ex vivo, they actually show different properties, especially in the exchange time, which is much shorter in the ex vivo settings. What finally has been proposed as SendDX is to account for both this contribution, both the cell body and the exchange, because this seems to be it seems to be the three most important features for gray matter to account into your model seems to be the cell body, the fiber as sticks, and exchange between the intranucleate and extracellular space. However, such a complex model raises challenges in terms of degeneracy and in terms of information to encode into your measurement. And as we discussed in one of the questions previously, the way you acquire the data is important and need to be suitable for the kind of modeling you want to do. And there is work ongoing in how we can optimize acquisition to encode enough information to have all this information estimated precisely and accurately. And so with this, I conclude with a few take-home messages. The design of sensible biophysical models for gray matter, it is critical to design these models. It's critical to know what are the meaningful microstructural features that we can measure, and that the complex tissue composition and water exchange across the cell membrane seems to be extremely important in gray matter, and we should account for that in the modeling. However, this is quite challenging. And in general, the validation is also quite challenging because features such as the cell membrane is quite difficult to pin down with direct measurements from histology. And we are still seeking as a whole, as a community as a whole, what is the most parsimonious model of diffusion in gray matter. What we seems to have agreed is that it should account for water inside the soma, inside the extracellular space, and inside the neurite, but also it should account for some sort of exchange between these compartments. And with this, I would like to thank you all, and happy to answer any questions.
That was an amazing talk. Thank you so much. In the interest of time, we can have like one or two questions. Olivier, is there any question?
Yeah, I do have one question. So it's about resolution. So with the typical, diffusion MRI acquisition being 2 millimeters isotropic and the cortex being between 2 to 4 millimeter thick, we not only need to increase the modeling accuracy like you did, but to also increase the typical resolution at which diffusion MRI is acquired to better investigate cortical microstructure?
Oh, this is a fantastic question. Yes, I didn't have time to go into this, but this is extremely important, especially for human studies. Yes, all the work that is going on now, trying to acquire efficiently with high SNR, high resolution images, using spiral readout or using a more fancy acquisition, is super useful for gray matter. And it's really something we need in the community, besides the modeling and the measurements in the encoding schemes. We also need techniques that can enhance the resolution. Partial voluming, it's terrible. And that's why a lot of the studies at the moment in gray matter are done in preclinical, in rat or mice, where you have a very thick layer of gray matter, far from any partial voluming, and you can use relatively big voxels there. But to go to human, we need high resolution. We need at least 1.5 isotropic.
Great, thank you. Yeah, I think for the interest of time, we do have a couple more questions, but again, keep those in mind for the end session. I think we'll move on to the next speaker.
Thank you very much.